Okay, hello, I'm Megan, and I worked this summer on developing a diagnostic strategy to determine the frequency of an autism-related mutation in the gene called HERC2, and I worked on this with my supervisor, Barb Triggs-Raine. So the purpose of my research, a patient at the Cleveland Clinic was diagnosed with a single base pair mutation at position 4625 in the coding sequence of this gene, and it was a G2 and A. This patient was of Ashkenazi Jewish descent, and Barb had previously acquired many of these DNA samples from the Ashkenazi Jews, so we wanted to screen all of these samples for this mutation to see the frequency of it. And we were also given a patient sample from Barb's colleague Marvin at the Cleveland Clinic, so we wanted to confirm that this patient did have the mutation that we were looking for. So the patient DNA we were given, this was all the diagnoses he was given, and he was diagnosed by Dr. Marvin Neufeld at Cleveland Clinic. So he was 19 years old, he had autism, cognitive delays, sleep disorder, delayed digestion, toe walking, and he was homozygous for this mutation we were looking for. And all these phenotypes indicate that the mutation has a neurological impact. So a little bit about the gene HERC2. HERC2 stands for HECT, an RLD domain containing E3 ubiquitin protein ligase 2. So it is an E3 ubiquitin ligase. So just a bit about ubiquitin. It is a small regulatory protein involved in post-translational modification. And it affects the protein in many ways, but the main way is by signaling for its destruction via the proteasome. So ubiquitination is carried out by E1s, E2s, and E3s, and since HERC2 is an E3, it is part of this process. So ubiquitin ligase is a protein that recruits an E2 conjugating enzyme which is loaded with ubiquitin, and it is involved in ubiquitination by catalyzing the final step, which is the transfer of ubiquitin from the E2 to the substrate. So you can see in this diagram that this is HERC2, which is the E3, and then it takes the ubiquitin from the E2 to the protein substrate, and then it can go to the proteasome for degradation. And then since HERC2 adds ubiquitin on, it can interact with many proteins that it is adding the ubiquitin to. Many of these proteins are associated with cell trafficking and energy metabolism, and because it is capable of interacting with so many proteins, some specific substrates are involved in causing neurodevelopmental delay because of decreased synaptic transmission. So some of these neurodevelopmental diseases are autism, Prader-Willi syndrome, and Angelman syndrome. So HERC2 has previously been associated with autism, and the patient of Ashkenazi Jewish descent was found to be homozygous for this mutation. This is exciting because it could raise the possibility that this mutation could be common in the population and possibly found in other people with autism as well outside of this population. Prader-Willi syndrome and Angelman syndrome have also been found in other studies, and these diseases are very similar to each other. In Prader-Willi, the maternal copy of the genes is silenced where the paternal is mutated and non-functional, and in Angelman vice versa, where the paternal is silenced and the maternal is mutated and non-functional. And then going along with the developmental delay phenotype that's seen in mutated HERC2, a mouse gene was made with a HERC2 knocked out, and the mouse also showed severe developmental delay. So this is the mutation from the G to the A, so it's located in the middle of the codon, changing the amino acid from an arginine to a histidine. The patient's DNA was sequenced, and this was the only significant change found in the gene associated with autism. And HERC2 is highly conserved across the species, so the arginine is also present in mice, indicating that it probably has a conserved role in the cell. So this is the gene structure of HERC2. It's located on chromosome 15 at position 13. So this is a bigger picture of the whole chromosome, so it's located where the arrow is. And then this is a zoomed out picture just showing it right there and like all the other stuff around it. So it is a fairly large gene as it spans 250 kilobases and has 93 exons, which is pretty big for a gene, one of the biggest ones. And it also has pseudogenes, which are non-functional genomic DNA sequences with very similar sequences to the true gene. However, they have no ability to express or code anything. So there are about 10 pseudogenes that are very similar to this gene, and they are found on chromosomes 15 and 16. So this picture, don't worry about all the little details. You can just look at the red stuff. So this is the mutation where it's located, and this is the true. This is the true HERC2 gene, and that's where the mutation is located. And then I just showed you some other pseudogenes around the area. There's other ones that are farther away that wouldn't fit in the picture, but this is just for you to get an idea. So in order to identify between the wild type and the mutant, we had to create a restriction site. So we used the enzyme NLA3, which cuts after the sequence CATG. So if you look at the mutated HERC2, it has the sequence right here, CATT. So we introduced a G to make it CATG with primers. And then the NLA3 would cut the mutated at CATG. And then the wild type, it would be CGTG, so it wouldn't cut. So then this is showing you the sequence with all the pseudogenes. So this G is where the A would be in the mutant. So this HERC2P2 is a problem because it is the same sequence as the mutant. So if it cuts, if it amplifies in the PCR, it could cut, and we can mistake it for the mutant when it is really just the pseudogene that's cutting. And then this is showing you the bigger picture of the sequence with all the pseudogenes. So the yellow is the primers that we used, and the blue is the mutation. So in order to try to only amplify the true gene, we introduced these C and T mismatches to destabilize the primer, hoping that it would only bind to the sequence it matched the closest with, which was the true gene. And then this is just showing you the G that we put in to make the CATG NLA3 site. And then when we use this strategy, this is what we got. So this is good because this is what we wanted. So the wild type didn't cut even when it was put with the enzyme, and then the positive control with the mutation did cut. And then we sequenced the DNA from here, and the wild type shows a G right here where the arrow is, which is good. And then the positive control shows an R, which means that it is a purine, so guanine or adenine. And this is okay because our sample was a heterozygote, so it could have the G and the A at that position. And then we used this method on some extra DNA in the freezer. And when we went to use it on the Ashkenazi Jew DNA, NL3 cut all the samples. So the preparation of the samples must have been slightly different because the pseudogenes amplified a lot more in the Ashkenazi Jew DNA samples. So we had to use some more optimization strategies. So I presented earlier the primers that I had used, but we tried many different combinations, different locations in the genes, adding more mismatches, less mismatches. So we did a lot of optimization with that. And then we, things like the techniques that seemed to work the best were decreasing the cycles, increasing the magnesium concentration to help stabilize, and increasing the quantity of DNA to provide more template for the PCR reaction. So when we tried this method, this is the picture of a gel that we got. So these are just the numbered samples. And then this is the positive control. So you can see that there's two pretty strong bands. And then these bands are all weaker, the ones that are cutting. So we just assumed that the pseudogene was there a little bit. But we classified those all as not having the mutation because they look slightly different. And then on this gel, we think that there might be a true positive on number 37 because it is a bit stronger there, and it looks closer to the positive mutation. So for the conclusion, we confirmed that the patient DNA did have this mutation that we were looking for, And so far, we've determined the frequency of the mutation in the Ashkenazi Jew DNA samples is around 1 out of 150, and we're still working on amplifying the rest of them and determining that. And finally, I'd just like to thank CRIM a lot for funding this great opportunity that I've had, and also for Barb's colleague, Dr. Marvin Natovich, for providing all the DNA, and also the University of Manitoba for the facility, and Barb and Jeet and Rick for helping me out in the lab. Sure.
First of all, my condolences that you took a gene with so many situations. I'm experienced that. It's not nice. But one question I have is just kind of a general question, is that ubiquitinolipase is probably one of the most common genes in the gene with multiple family members in there. Yet there are specific mutations in this one type of gene. Has anyone speculated as to why this particular is so important?
I don't think so. Not that I know of. Just like the reason we studied it was because this was where the mutation was found in the patient. So other than that, I'm not sure.